Hi everybody, welcome back to another review video. This one's going to be all about RNA processing and transcriptional control. Now I apologize, there's going to be some information that I was going to teach a little bit later, but I actually moved it into this video because I think that the flow of information is just a little bit better. And so yes, I changed the order of your notes. I'm sorry, forgive me, but I promise you it'll maybe make a little bit more sense and I think it'll be worth it. So if you watched my last video, you should now know that the primary product of transcription is RNA. And so this video is all about RNA processing, so we're going to get a little bit more information in there. But the primary product is RNA, and we really pay attention a lot to mRNA, but there are other types of RNA. Now, we spend a lot of time talking about messenger RNA, but we also have ribosomal RNA, aka rRNA, and transfer RNA, aka tRNA. And these will both become very, very important when we get into translation. But for right now, I'll just let you know that rRNA actually sits inside the ribosome, which remember, if you remember back in Unit 2, the ribosome is basically a big protein complex, well, two subunits of big protein complexes, and running between these two is an rRNA molecule. And this rRNA molecule, surprisingly, acts somewhat like an enzyme. In fact, it acts a lot like an enzyme because it catalyzes peptide bond formation, which sounds like a lot of big words, but it's catalyzing the reaction that puts two amino acids together. It forms that bond. And without that rRNA, it wouldn't be possible. And so it's not an enzyme because it's not a protein, but it has catalytic activity, so sometimes people refer to it as a ribozyme. Now, transfer RNA, again, we'll talk about this more in detail later. It is going to bring those amino acids that are going to get joined together to form a protein, is the thing that brings those to the ribosome. So that's the most important pieces, and we'll get more into it when we get to translation. But the other piece I want to show you here is that with rRNA and tRNA, they have these kind of funky shapes. So everything shown in blue here is the RNA molecule. And it kind of looks like a weird sort of alien-like snowflake. And maybe you remember in class that the reason why is because these flat regions here, and these flat regions here and here and here, that was my cat. Those flat regions are going to be where there's complementary base pairing within the same molecule. So right here, it might be CAT. And if you follow this line down, it's still the same molecule, still the same strand. This was C up here, so this might be a G. This was an A, so this might be a T. And I really should say U because we're talking about RNA. And then instead of a T here, so a U, then this would be an A. And so we can have complementary base pairing in the same molecule. This does not mean it's double-stranded because it's still just one single strand, but we do say that it has secondary structure. So rRNA and tRNA have secondary structure that gives rise to new function. Because remember, structure dictates function. We see it all the time. Okay. Now, we are also going to be focusing, when it comes to RNA processing and transcriptional control, we're really focusing on eukaryotic gene expression here. And we really have to understand the mechanisms of transcription and translation in both of the different cell types to see why. So if we look at the prokaryotes, well, their DNA is just floating around. There is no nucleus. That's very characteristic of a prokaryote. So there's no nucleus, which means that the ribosomes have access to the mRNA basically as soon as it is synthesized. So transcription occurs, and as soon as that RNA is coming out, there is ribosomes right there. And those ribosomes are going to start translating immediately, which means that there is literally not time for much processing of this RNA. So when we're talking about RNA processing and transcriptional control, it's very, 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 very limited in prokaryotes. Whereas in eukaryotes, transcription happens in the nucleus. So we have pre-mRNA that is made directly from the DNA, but that's not a finished product. Instead, there's a lot of processing that has to happen here to get to a mature mRNA. And then once it's all mature, that's when it is going to leave the nucleus, and ribosomes are going to come in and start translating. But because we have sort of this, I don't want to say protective, but somewhat protective barrier for this mRNA, there's a lot more steps that can be involved, and we can do a lot more processing, which is sort of an efficiency thing and an advantage for eukaryotes. And then only after a while, after we've done these changes, these modifications, is it going to leave the nucleus to be translated. Okay, so now that we've laid that foundation, let's start talking about it. The first post-transcriptional modification is the addition of the 5' cap and the 3' poly A tail. So that sounds like a bunch of words, but let's break it down. So on the 5' end of this mRNA molecule, we add a GTP, a guanosine triphosphate cap. And that is just going to help the mRNA molecule attach to the ribosome. On the other end, the 3' end, we add a poly A tail. Poly meaning multiple, A for adenine, so just a bunch of adenine bases here that are going to help the RNA be more stable and ensure it doesn't get degraded. Now, the other really important part of mRNA processing is splicing. So, what is splicing? Splicing is the retention of exons and splicing out of introns. To really ensure that you know what this means, there are regions in the DNA that are somewhat considered junk. That's the introns. Now, we don't really believe they're junk. There's probably some use for it considering they're conserved, meaning that they are found in lots and lots of different organisms, and there's some sequences that are conserved, which indicates that perhaps there is some evolutionary advantage. But we don't know what they do. And we do know that they don't make it into the final mRNA, which means that they are not ever going to be translated. So those are the introns, and they are going to be spliced out by a protein complex called the spliceosome. So the spliceosome chops out these introns and it glues together exons. So the exons are what make it into the final mRNA molecule. So with those changes, those modifications, we now have a mature mRNA that is different than the pre-mRNA, which was just what the gene said to transcribe. Then we have mature mRNA that has the addition of the 5' cap, the poly A tail, and only has exons left. Okay. Now we also can have something called alternative splicing. And this kind of throws a wrench in that central dogma. And I'll talk about that in a second. But looking at this image here, you have chromosomal DNA. And your RNA polymerase can read, and maybe the terminator sequence is right over here after exon 6. Well, if we just went with regular splicing, it would have all 6 of those exons. But in reality, that's not true. Usually, and in a lot of genes this is the case, there are alternative splicing patterns. And so in this first protein, it retains 1, 2, 4, 5, and 6. So it chopped out exon 3. For this second one, it chopped out 2 and 4. And for this third one, it chopped out just 2. Now, this is always going to result in different proteins, right? If I have a different sequence, that means it's going to bring different amino acids. Different amino acids mean a different protein. And so this kind of throws a wrench into what people thought was the original way the central dogma works. There was sort of this idea that there is one gene, one protein, and so this would be one gene, but it actually results in 3 proteins. And this is actually supported. We think that there's about 100,000 proteins in the human body, roughly, but only 20,000 genes. So that is in part due to alternative splicing. This is one of my favorite things. You should absolutely expect a test question on this because I think this is absolutely incredible. So neat. Let's keep going. Okay, now I want to start thinking about regulation. So would it be good if all of our genes were expressed all of the time? Now, anytime I hear words like all, that should automatically raise alarm bells. No. The answer is no, it would not be good for all of our genes to be expressed all the time. If I'm starving, it would not make sense for my body to express genes to store fat, because I need to oxidize that fat. I need to get that moving so that I don't die. If I'm storing that fat instead of burning it for energy, I die. And so, no, it would not be good for me to express all of my genes all the time. Sometimes I need to burn fat. Sometimes I need to store it. It just depends on my environment a lot of times. So, how does our body know? How does our body know when to express and when not? Well, there are a lot of cellular mechanisms that we are going to talk about. For eukaryotic regulation of transcription, there are three main areas that we talk about. The first two are really relating to this first number here from DNA to RNA transcript. These first two are really saying, okay, how do we regulate whether or not this transcription even occurs? And so the chromosome is really important for that, which we'll talk about in a second, and definitely transcription factors are also extremely important for that. But once we already get the RNA transcript, then processing control and also degradation control become really, really important as well. And so understand that just because we got the transcript, just because we matured that transcript through the modifications, that does not necessarily mean that is going to become a protein, because we have other mechanisms in our cell that is going to regulate it even after transcription. So this first one, talking about chromosome structure, you're really going to have to know the structure of a chromosome. And remember, a chromosome is not just DNA. We often talk about it like it's just DNA, but that's ignoring a really important element of the chromosome, which is protein. And DNA really is wrapped around a bunch of proteins called histone octamers, or just histones, usually people say. So these histones have a partial positive charge. They're a little bit positive because DNA is a little bit negative. And if you don't remember why, it has to do with that phosphate group in the nucleotide. So remember, a nucleotide is phosphate sugar base. That phosphate group is slightly negatively charged, which means that overall DNA has a net negative charge. And that means that it needs to have some positive proteins to wrap around. And that's really why we have to have DNA in a chromosome structure, because otherwise it'd be highly unstable with that negative charge. Now, I want you to think back in class when we had that pencil and yarn example, when we have DNA that is really, really tightly wrapped around histones, those genes are not going to be as easily accessible. And so this is one way that we can sort of regulate our genes. And so this deals with literal access to the genes. If DNA is wrapped really tightly, if you're following my pointer, I hope you are, if it's wrapped really tightly around a histone, these genes are not accessible. So they are going to be really hard for RNA polymerase to transcribe, and then that transcription product would end up going to the ribosome, right? But I can't do that if it's wrapped really tightly, whereas this is accessible. So what happens is these histones can move. They can move and they can rotate and unwind the DNA, and then they can come back. That is one way that we can access the DNA. Another way is through epigenetic modifications. And the two most famous epigenetic modifications are methylation and acetylation. So methyl groups usually condense DNA around histones, so they wrap it a little tighter. So if a cell really does not want to express a gene, it might heavily methylate a specific area to condense it around the histone. Whereas acetylation is going to loosen the DNA around histones and make that DNA more accessible for RNA polymerase to transcribe. Now, these are the foundation for epigenetics. So epigenetics, epi meaning surface, genetics meaning pertaining to genes and heredity. These are going to control which genes are expressed through environmental factors without changing DNA sequence. And we actually have a little bit of evidence to show that epigenetic modifications can actually be inherited too. So things like methylation and acetylation might be inherited from from parents to offspring, which is pretty cool. And I strongly recommend you guys go in and research that because there's lots of fun stuff. So this is the foundation for chromosomal control of gene expression. That said, although that's my favorite, I think that is the most interesting stuff, there is also a lot of transcriptional control, which is arguably more critical, and it certainly provides the most control. Probably the most important part of this is transcription factors. So transcription factors, hopefully you remember, they are proteins that influence RNA polymerase's ability to bind. And they are so important, and the transcription factors largely determine cell type. And I'll talk about that in a second. There are also regions of DNA that are going to encourage certain transcription factors to bind and RNA polymerase to bind. These are other regulatory regions called the enhancers and silencers. When we talk about the lac operon and the trip operon, this can be a little bit more clear, but for right now, just understand that they are regulatory regions that are going to either activate or repress transcription. Okay, now this is what I was saying with transcription factors varying by cell type. So in large part, what makes your liver cell a liver cell is the availability of certain transcription factors. So in a liver cell, you have these transcription factors that lead to these genes being expressed, which lead to these functions. So metabolism of drugs, synthesis of blood proteins, and lipid storage. The osteoblast is a bone cell, right? They have these transcription factors, which lead to these genes, which leads to bone formation and mineralization. So without these transcription factors, every cell has the same DNA, but how does it know which genes to express? Well, it's the availability of certain transcription factors that are going to tell the cell to be that cell. And this is super important in development. So the induction of transcription factors during development is going to result in sequential gene expression. I really like this image. That's why I printed it out for you and I'm giving it to you in class. But it's really important to understand that these transcription factors are not only important in maintaining that cell type, but it also is important in differentiation and development. And it's very important. So this last piece, like I mentioned a few slides back, really has to do with this mRNA degradation control. We have the ability to degrade mRNA to ensure that it doesn't become a protein. Generally speaking, this is called RNAi or RNA interference. There are enzymes in RNA that interfere that will trigger the degradation of that RNA. The mechanism, we'll look briefly at it on the next slide, but you are not responsible for it. But I want you to understand that just because we have the mRNA, even once it's matured, that does not necessarily mean it's going to become a protein. There are other ways to regulate this and prevent it from actually becoming a protein. We also have to consider the lifespan of mRNA. mRNA is typically shorter in prokaryotes, but it can stick around a little longer in eukaryotes, and we can sort of preserve it and save it for when we need it to be a protein. That's not really possible in prokaryotes. So let's look briefly at this RNA interference. So in RNA interference, you have RNA that enters, and essentially what happens is you have these small interfering RNAs, that's what this SI stands for, that sort of matches and base pairs with your RNA, and it will knock out that mRNA. Again, the mechanism is way beyond this course. If you're interested in it, it's actually a technique that you can use in the lab. I've done this. I used to knock out genes in algae, and so that was pretty fun. But the idea here is that you introduce an interfering RNA, and that will tell the cell essentially to degrade a piece of RNA that you don't want it to be there. So then it won't express it. If that's totally bonkers, if that's totally out of the blue for you, don't worry about it. Just understand that even though we have a mature mRNA, it does not necessarily mean it's going to become a protein, because there are mechanisms that exist that can degrade it before it gets to that ribosome. Okay, So in summary, and this is not what you should necessarily put in your summary box, because your summary box should be complete sentences, you should know the modifications that mRNA transcripts go through to mature, and then you should also know how transcription and post-transcription is regulated. So you should know about the addition of a poly A tail and a GTP cap. You should know about splicing. You should know about alternative splicing. You should understand how chromosome remodeling, aka the movement of histones, can regulate gene expression, and epigenetic modifications, how that can modify gene expression. You should also understand transcription factor availability and how that relates to cell and tissues. And you should also understand interference after the fact. I hope that this was a little bit more comprehensive. If you were a little confused in class, I hope this clears things up. Good luck studying, and I'll see you soon. Bye.